Okay, Assalamu alaikum everyone. My name is Ayat. I'm a second year medical student, and today we're going to be talking about nucleotide metabolism, which is one of your last lectures in MOL. So that's nice. Um, First of all, we're going to be talking about the structure. I think we've been over the structure thousands of times throughout MOL and even genetics. So you have nucleotides, you have bases and they form nucleotides, and the nucleotides form DNA, they condense, they form your chromosomes. So we know about all of that, right? We The most important thing here is that DNA or RNA, both of them, they have sugar, they have ribose sugar, they have phosphate, and they have nitrogenous bases. Now these nitrogenous bases are our purines and pyrimidines. Um, And we know the names. We know adenine, guanine, thymine, right? There's two types of things that you should know. There is nucleoside and there is nucleotide. So a nucleoside, it has an S in it. I don't really know I mean, if you know nucleotide, you automatically know what a nucleoside is. But if you forget, in my head, S is like second, so it's like two for some reason. It's like second, so two. There's two things. There's sugar and base. Nucleotide, there is a T, so there's three things. There is sugar, base, and there is phosphate. Okay, so the phosphate is like the key difference between a nucleoside and a nucleotide. Okay. Um, so these are our purines. Both of them are our bases. They're both bases. These are our purines, and these are pyrimidines. Um, there are certain reactions that take place. You don't need to know the reactions, but when you remove the NH2 from adenine, you get guanine. When you remove the NH2 from cytosine, you get uracil, and then when you further methylate it, you get thymine. So those are like the basic reactions. Again, you don't need to know those. But to remember which ones are purines and which ones are pyrimidines, the doctor itself, she gave us these mnemonics. Pure as gold, so P purine, pure purine, as adenine, A, G guanine, gold. If you remember purines, I don't think you really need to remember the mnemonic for pyrimidines. But it's like cut the pie, cytosine, uracil, and thymine. The thing with pyrimidines is uracil is only in RNA, while thymine is only in DNA. So, and cytosine is in both DNA and RNA. So that's one thing that you should remember about them. Okay, next. So we said a nucleoside is a sugar and a base, right? So how do you name them? Okay, so purine nucleoside. Adenine is our base. All right. When you add a sugar to it, it turns into adenosine. So if I say adenosine, it's a purine nucleoside. Now for pyrimidine, first of all of the pyrimidines, after when they've become a nucleoside, they turn into dene. It's pretty obvious from the pyrimidine itself. That's why at the start when I said thymidine, I stopped and I changed it again because thymine is the base and thymidine is the base and the sugar. So that's one thing. All right. Okay, after the nucleoside, we have the base, we have the sugar. Now we need to add our phosphate. When we add our phosphate to this, again, when we add our, one second. Okay, when we add our phosphate to this, it turns into AMP, GMP. Now it depends on how many phosphates you add to this. If you add one phosphate, it's monophosphate. If you add two phosphates, it's ADP. Diphosphate, if you add three phosphates to it, it's ATP. And we all know ATP. Pyrimidine, if you add one phosphate again, it's the same thing. Another thing to know is that all of these phosphates, nucleotides basically, they're all used in RNA. So you have AMP, GMP, all of these are going to be used in RNA synthesis. What's going to be used in your DNA synthesis is DGMP, for example. So it's deoxy and then the and then guanosine monophosphate. Um, as I said, in DNA you're gonna have DMP, DGMP, and basically everything the same thing, but with a D because it's deoxy. Okay, you remove one of the oxygens. Yep. Um, okay, then you have these new names. They're like new characters unlocked in MOL all of a sudden. You have inosine, you have xanthine, you have hypoxanthine, and you have uric acid. Now, when I did this, I thought these names were like pretty cool. They are pretty cool. These are basically what's going to form your purines. So you've never heard of them before until now. So it's pretty cool, I think. Then you have nucleotides. We're gonna start the synthesis of nucleotides. One thing before I do that, I just said that these are new, right? Inosine, xanthine, hypoxanthine, and uric acid. What are they exactly? Now, for example, I'll take the example of adenine because we're all very familiar with adenine itself, right? Um, so if my base is adenine, right, and I add a sugar to it, it's gonna give me the nucleoside adenosine, right? And then I'm gonna add a phosphate to it, and then I'm gonna make AMP. This is like my basic recipe, right? I've been doing this for years. Now for inosine or IMP, what I'm going to do, here the base is hypoxanthine. And then when I add a sugar to it, it forms inosine. All right, these are two different names. Unlike adenine, adenosine, you have hypoxanthine and inosine. So they are related to each other. Mystery solved. IMP. Okay, you just add a phosphate to it, you get IMP. Okay, now first of all, we have PRPP synthesis. Now PRPP is like the boss in this whole game, alright? It just goes, it helps in the de novo synthesis, it helps in the salvage pathway, it's everywhere. Okay? And for PRPP, we need something called ribose 5 phosphate. How do we get ribose 5 phosphate? I'm pretty sure you guys have all heard of ribose 5 phosphate. We get it from the HMP shunt or the pentose phosphate pathway, PPP, which we have done, I think, in MOL 1. You guys took it in MOL 1. You have the HAP shunt, you get ribose 5 phosphate. Now, coming to mol 2, what do I do with the ribose 5 phosphate to make it useful in mol 2? I am going to turn it into PRPP. There's an extra P here. I'm going to turn it into PRPP, which is activated ribose sugar. I basically added phosphate to this. So I used an enzyme called PRPP synthetase. So whenever you see synthetase, there's a T there. It means that it uses ATP. All right. And ATP turns to AMP means I take the phosphate and I added the phosphate here. So activated ribose sugar or PRPP is just sugar and phosphate. That's what it is. All right. Now I have my sugar and phosphate, and I need one last thing. I need the nucleotide. Sorry, I need the base, right, to form the nucleotide. So how do I get the base, right? Now in purine synthesis, what happens is this is like this is my PRPP, okay? And now I'm gonna keep adding ingredients here, like atom by atom, and I'm gonna start forming the base ring. All right, we'll come to that. Coming here again, we have this is an inhibitor, purine ribonucleotides, obviously, because if you have enough ribonucleotides, why would you want to continue with the pathway and waste your own resources, right? So you just stop the whole pathway. You stop this step here. In inorganic, uh, sorry, how do you increase more synthesis when you have more phosphate? More phosphate you have, more product is formed. Okay, now you have your PRPP. This is the main thing that you need. PRPP activated ribose sugar, uh, sugar plus phosphate. In purine synthesis, the base is made from scratch, bit by bit, on PRPP. Okay, this is going to be one of the main differences between purine and pyrimidine. In pyrimidine, you're going to make the base ring first, like the hexagon ring, and then you have PRPP, and then you're going to be like, wow, unite with each other, you know? This is the difference. Here you're gonna make the ring, and then you're gonna attach a thing. Over here you made PRPP, and then one by one you're gonna make like, okay, one and then two and then three and then just some atoms together, and then you're gonna make the ring on top, basically. Okay, so purines are not synthesized as free bases. All right. What do we need for a purine ring? We need amino acids, okay? Specifically, a mnemonic called GAG. Okay, you need glycine, you need aspartate, and you need glutamine. Now, what does this give you? This gives you all of the nitrogen that you can see here: nitrogen, 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 nitrogen. Right? Then you need sugar. Okay, this ribose phosphate comes from the HMP shunt that we just said, the PPP pathway. That's the sugar. And then you need tetrahydrofolate (THF) and carbon. Okay. Tetrahydrofolate. The thing with folic acid in humans is that it's not produced in our body, so we take like supplements for it, or we take it from food, the food we eat. Okay. Basically the diet. Okay, this is one of the most important steps, although I keep forgetting about this. That's funny. Okay. Committed step. This is the committed step. So once you perform this step, there is no going back, right? You have to make purines. Another thing is that it's the regulated step. So when you make the purines and they're like enough, all right, we're enough, it's going to shut the entire pathway down. So it's like the regulated and the committed step. What happens in this is that first you have the glutamine and glutamine, and we said the glutamine amino acid is for nitrogen. It's going to give us the nitrogen, all right? Basically, it's going to give it to us. It's going to leave, and then we have the water. Sorry. Okay. The glutamine, and then you basically have something called phosphoribosylamine, right? The amino group. Phosphoribosyl. P. If you break the name down, you get it. Phosphoribosyl sugar. Because we said that PRPP was an activated form of ribose, so it was P plus ribose. Right? Now you took glutamine and then you added the NH2 group here, right? So that's phosphoribosylamine. All right? That's how I remember it. You just have to break the words down and you get it. Okay, the inhibitors are AMP and GMP. Again, I think in every single step, the inhibitors are going to be AMP and GMP. Why? Because if you have enough at the end, then you don't really need to make anything else, right? So that's why they're the inhibitors in every single step, almost. The activator is PRPP because if you have more PRPP, the reaction is going to go forward, like equilibrium, right? So yeah, if you have more stores of PRPP, you get more phosphorib, more substrate, more product. Again, phosphoribosylamine. Wait, let me just delete all of this. Okay, phosphoribosylamine. Phosphoribosyl is our PRPP, basically. And then you added an NH2 to it. Where did you get it from? You got it from glutamine. And this is where the amine comes from. So phosphoribosylamine. And this is the committed and the regulated step. Remember that. And then we skip step 3 to 11 because they're not important. Uh, what happens is after you form the phosphoribosylamine, you form something called IMP. Okay? IMP is important, but it's also our parent nucleotide. So it's gonna give you AMP and GMP. And there's also something called N10 formyl DHF. Don't need to know this, don't need to know this. Just remember it's DHF, alright? You just need that in steps. And we talk about this here because there are certain medicines that target DHF. So we're gonna talk about that later. Okay, so we had ribose, right? After we had ribose, we turned the ribose to PRPP. When we made PRPP, we added an NH2 from glutamine and then we made it to phosphoribosylamine. Alright? Then phosphoribosylamine, we skipped like step 3 to 11 and then we got IMP. right? Okay, now we're at IMP. IMP is supposed to give us both AMP and GMP. Okay. Uh, I don't think it's pretty clear here. Hmm. But all right. Wait. Okay. This enzyme is IMP. Let me just write it down. Dehydrogenase. GNase. Okay. This one is IMP dehydrogenase, and this is a focus here because we have a drug here again. But anyways, so you have IMP. IMP goes and then eventually forms AMP. All right. Forms AMP. Again, because AMP is the end product, and if you have enough AMP, it's gonna go and it's gonna stop its enzyme, right? Like stop making me like I'm enough, right? Another yeah. Okay, this is basically feedback inhibition. Over here, you form GMP. Okay, with IMP dehydrogenase, you form GMP. And again, if GMP is enough, it's gonna feedback inhibit its own enzyme. They're not gonna go and inhibit each other. The different it basically AMP is not gonna go and inhibit IMP dehydrogenase. It's gonna go inhibit its own enzyme. All right, that's one thing. And the funniest thing about this is they're kind of dependent on each other because you see, you form GMP here and eventually you're gonna form GTP, right? When you're making AMP, you need this GTP, and when you're making GMP, you need ATP. So that's kind of a funny thing how they're like codependent on each other. All right. We're gonna talk about this drug later. Again, you have IMP. IMP. Right. Okay. IMP forms GMP, and then it also forms AMP. Okay. And the important enzyme here is IMP dehydrogenase. Okay. And they feedback inhibit their own enzymes when they're enough. That's basically the pathway. If we were to go over it, there is HMP shunt, and then you have ribose. You have ribose 5 phosphate. Okay, and then after ribose, it's better if you guys draw with me. Whoever is watching this, ribose 5 phosphate, and then you turn it into PRPP. With what enzyme? With PRPP synthetase, right? PRPP synthetase means you're synthesizing PRPP, and T means you're using ATP. Um, this is the activated form of ribose. So you have sugar, and you have take it and make the phosphoribosyl amine. All right, and then here you use the mitotransferase enzyme. Did we talk about the mitotransferase enzyme? 1 second. Yeah, this is aminotransferase because you're transferring the NH2 transferase. All right, that's basically it. Okay, PRPP, and then you have phospho blah, 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 aminotransferase, and then you have wait, amine, and then you form 3 to 11. You skip these steps. You form IMP, and then you have IMP dehydrogenase, and then you form GMP, and then here you form AMP. That's basically the de novo synthesis, alright? Okay. Now, after we learn the de novo synthesis, I think it's easier to know what the drugs here do. Um, Methotrexate, this one has been repeated in like a lot of questions in the finals from what I've seen. Like, yeah, which step does it inhibit, and then what are the adverse effects, and all of that. So I would say this is important for you to remember methotrexate. Um, it's a folic acid analog, Analog is basically something that resembles the substance itself. So it resembles folic acid itself, but it's not folic acid. So it's like a doppelganger, just here to cause problems, right? It's like the evil doppelganger. It's going to interfere with nucleotide synthesis for rapidly dividing cells. So in our body, as I said, we don't form folic acid. Okay, we take it from our diet. So when we take it from our diet, then we go ahead and then we have that we have dihydrofolate, and then we have tetrahydrofolate. Now we have this whole step thing, DHF. Well, when it's gonna form the THF, there is gonna have a problem. It's not gonna allow it to form the THF, and the THF, we use it in our DNA synthesis in the whole de novo pathway, the 3 to 11 steps. There are a few steps, there's two steps that are gonna use the THF. Now, if you stop the THF, then you cannot reach to your purines. If you don't reach to your purines, you cannot make your DNA. So there is no DNA, so your cells can't form properly. That's why at high doses it's used. You basically cannot add the carbons in the DNA, and that's why at high dose it's an anti-cancer. Because if you can't form DNA, then the rapidly dividing cells basically they need the nucleotides, and you're not giving them the nucleotides, so they're destroyed kind of. So that's why it's an anti-cancer. At low doses, it's used for rheumatoid arthritis too. Again, folic acid analog. Okay, so it has certain adverse effects. Macrocytic anemia. There was a picture in one of the exams. I'm not sure if it was um for the sickle cell thing or it was for the anemia, but there was a picture that came from the slides, so it's better that you guys know. Macrocytic anemia is basically two types. There is megaloblastic and non-megaloblastic, and then there's non-megaloblastic. Took this in FND, non-megaloblastic. The megaloblastic one is the one that's of concern here because megaloblastic anemia is caused by deficiency of DNA, you could say. So you don't have enough DNA, and then eventually you have megaloblastic anemia because in your RBC lineage thing, I don't know if you remember, you had like before your RBC, you had the premature RBCs. All of them had DNA. So you're not giving them the DNA to form. Okay? And then that's why you have megaloblastic anemia. You don't need to know the whole thing. Just know that you have macrocytic anemia. You have scaly skin, you have immunodeficiency because your own cells are being destroyed too, right? And you have baldness. You have all of these because methotrexate just targets any or affects any rapidly dividing cell. All right, that's why you have baldness, immunodeficiency, anemia, and scaly skin because all of these they basically rapidly divide. All right, sulfonamide is a PABA analog, and the thing with this is it doesn't work um, exactly like it doesn't work exactly like methotrexate, so it won't exactly destroy you because it inhibits this one enzyme that's only present in microorganisms. So even if you take sulfonamides, it's not going to affect your own cells because you don't have the enzyme. So it's basically not interfering with your own process. Okay, that's so this is basically an antibiotic, only affects microorganisms. Then you have mycophenolic acid. This is the one we talked about over here when we were forming the GMP and the AMP. This one goes and inhibits IMP, goes and affects IMP dehydrogenase. So you don't have GMP, you have no purines. And then you know how that thing I told you where you need the GTP here. So if you have low GMP, you eventually have low GTP, and then you have low GTP, and then you have low MP. So this is like a whole cycle that's going on over here. So this is used in immunosuppressant because T and B cells, the T and B cells of our immune system, they rely heavily on de novo synthesis. We have two types of pathways. We have de novo and we have salvage, right? So T and B cells only rely on de novo synthesis. So because we're gonna stop the GTP production, they're going to stop proliferating. And they have no other way to get the GTP because they don't rely on salvage, right? They only rely on de novo. That's basically it. It's a uncompetitive inhibitor of IMPDH needed for the de novo synthesis of purines. And they rely heavily on de novo synthesis because they're rapidly dividing. That's basically it. Hmm. These are all of the drugs that were used in the de novo synthesis. You have the one we just talked about, mycophenolic acid. There's another one. It's called ribav- ribavirin, ribavirin, and it also inhibits IMP dehydrogenase. This one is you don't need to know much about this. Just know that it's an antiviral that also inhibits IMP dehydrogenase. Then you have sulfonamides and you have trimethoprim, which was in the slides but she didn't talk about. And the main one that you have to remember is methotrexate. So don't forget about methotrexate. That's like the main one. Then you have the purine salvage bottle. Okay. From the word salvage, what do you think salvage basically means? Salvage means to kind of like save something, right? You're gonna take whatever from the cell turnover when the cell dies. Yeah, the scraps. You're gonna take the scraps of whatever is left, and you're gonna take that and then recycle them, and then again make your nucleotides and then use them in your DNA again. All right. So 90% of our purines are salvage, so that's a good thing, right? 10% of it turns to uric acid. Uric acid was also one of the things that we saw when we saw the inosine sampling thing. 10% of them are uric acid. Uric acid eventually they go to urine and we excrete them out, basically. We're gonna get our bases back. We're gonna get adenine, guanine, and hypoxanthine. All right. Um, okay. This is basically if you forget what hypoxanthine was, I put it here again. So hypoxanthine is the base for IMP. Okay. How do we get the bases back? Okay, you have the base. You have adenine, for example. I don't know. I think adenine is my favorite one. I think I just used that as an example everywhere. You have adenine, all right? And this is your base, okay? And in a nucleotide, you need a base, you need sugar, and you need phosphate. Now, to make this a nucleotide, we need to add sugar and phosphate. So throughout this entire thing, throughout purines, throughout pyrimidines, throughout everything, what we're trying to do is we're just trying to get three friends together the whole time. You need just sugar, you need phosphate, and you need base. They're all just trying to come together, basically. That's all you're doing throughout the lecture. You have base, you have the base, you have the sugar, and then you have the phosphate. Now, adding the sugar and phosphate to this is called phosphoribosylation. Ribosylation. Okay, so that's how you're gonna get the nucleotide again. Again, another thing, as I mentioned before, phosphate and ribose together is an activated form, right? And this activated form has a name, which is PRPP. It's back again. So PRPP is used here again. You have the base, you have PRPP combine, and then you just form the nucleotide all over again. Obviously they need enzymes. Nothing can ever work without enzymes in the body. For adenine, you have APRT, which is adenine phosphoribosyl transferase. I think that's pretty obvious from the name itself. You have adenine here, right? And then you have PRPP, right? So adenine on adenine, phosphoribosyl, the phosphoribosyl, you're transferring the phosphoribosyl to adenine. That's basically the meaning of the enzyme. So you don't really need to memorize the enzyme when you understand what it's doing. Okay? Then you have AMP, basically, because nucleotide, right? So this enzyme is not really as important as the next one because the next one works for two nucleotides. So the more the merrier does not stand in this condition. HGPRT, hypoxanthine guanine phosphoribosyl transferase. Okay. For guanine or hypoxanthine. Okay. So hypoxanthine for GMP, ignore the hypoxanthine here. The guanine. Guanine phosphoribosylate. Combine transferase. Transfer it here and then you get GMP. Here, ignore the guanine part. Hypoxanthine. Don't ignore it in the exam if you're writing it down. It's, for understanding this. Hypoxanthine, PRPP, you get IMP. Hypoxanthine, guanine, phosphoribosyl transferase. This is important because later we're going to talk about a disease, a clinical condition, I would say, that happens when there is deficiency of HGPRT. Okay. Another thing I mentioned before at the very start was all of these AMP, GMP, IMP, all of them are going to be used in RNAs, all right? For DNA, we have a completely different thing. We have not completely different, I would say. You have AMP, you have GMP, so you just have this D there, just because deoxy, alright? You have ribonucleotides, you have deoxyribonucleotides. But how are you turning the AMP to a DMP? You use something called a ribonucleotide reductase. Okay, so whoever's taken chemistry in school, we've done reduction and oxidation. Reduction means removing the oxygen basically. So you can see here there is OH here, but there's this H here. So you just remove the oxygen, right? So that's why it's ribonucleotide, because RNA, reductase, reducing the ribonucleotide to get your deoxyribonucleoside. So it's pretty obvious from the name again itself. Um, and here what inhibits it is DATP, because again, you have more DATP, then you don't really need the process to keep going, right? Why do you need so much DATP? That's why you're gonna, when you form the DATPs and there's enough DATPs, it's gonna be like, all right, stop, we're enough. Okay, that's basically it. All right, the drug here. There's another drug. There's a lot of drugs in this, honestly. I never realized that until I studied this again. Deoxyribonucleotides. You have AMP to DMP, DAMP, and you have something called hydroxyurea. Okay, don't get this confused with the insulin drug. There's sulfonamide is in this lecture. That one was sulfonylurea, and hydroxyurea is in this lecture. If you confuse it, I don't know, don't confuse it. Hydroxyurea. Okay, it's gonna affect the ribonucleotide reductase. Now your RNA cannot form DNA. If your RNA can't form DNA, then well, good luck surviving. Hmm. This is used. This is used to treat for sickle cell, which induces HbF. I think this was one of this may be the picture that was there. So I just put it here for you guys to see. It's pretty obvious. The blood is kind of shaped like a sickle cell, and it's anti-neoplastic. Again, it's anti-cancerous, because of the same thing. Most of the drugs here are anti-cancerous because, as we know, cancer cells they what do you say? They replicate, they divide very very rapidly. All right. So because they divide very very rapidly, they use the DNA synthesis thing. Now, if you stop their DNA synthesis, then they're not going to be able to divide. That's why we have a lot of anti-cancerous drugs in this, like methotrexate and hydroxyurea itself. It's anti-neoplastic. Neoplastic and cancerous is the same thing, by the way. Anti-cancer, anti-neoplastic. No DNA for cells to divide. Cells can't divide, and wow, we destroy the cancer cells. Um. Okay, so we formed the purine, and now we need to degrade it. Quickly going over it again one more time. Should I just go over? Okay, I'll just make it over here, honestly. Again, you have the PPP or the shunt. You probably won't ask you this, at least not in this lecture. Then you have the ribose 5-phosphate, and then you activate it. How do you activate it? You add phosphate to it, so it becomes PRPP. with PRPP synthetase. All right. Now this PRPP, you're gonna add an NH2 or PR phosphoribosylamine. All right. Amine. This is the committed step. All right. De novo. De novo means you're making it from scratch, by the way. This is the amine was the committed step, and now you have IMP. All right. You skip a few steps. You have folic acid. You have THF there. You have other stuff there. You skip a few steps. You go to IMP. Now with IMP, you have AMP and you have GMP. All right. With GMP, you have for forming GMP, you have IMP dehydrogenase. All right, and this is the target of a drug. All right, can you name the drug? Hmm. Yeah, that one. Uh, mycophenolic acid. Mycophenolic acid is gonna go to IMP dehydrogenase, and it's gonna stop the basically the GTP production, and eventually you're not gonna be able to form DNAs, right? That one. Okay, that was de novo. Then you have the salvage pathway. Salvage means salvage means scraps again. So you have all these scraps that you're trying to put together again because reuse, recycle. We love the environment, right? Environment in ourselves in this situation. Okay, so you have all of these bases again. Okay, and as I said throughout the whole lecture, you're just trying to reunite three friends. So you have the base adenine, you have the base adenine, and you want to add um phosphate and sugar to it. Now, what is phosphate and sugar called together? It's called PRPP. All right. Again, PRPP comes here to save the day. PRPP, adenine, you form nucleotide. All right. Okay. And uh, uh, also, it's here, so I'm just gonna go up here. Also, for adenine and PRPP, you're gonna use adenine phosphoribosyl transferase. Transfer phosphoribosyl to adenine. And for it, IMP and GMP, you're going to use AGPRT, transfer of phosphoribosyl to guanine or hypoxanthine, whichever one you like more. Um, and then you have this drug, hydroxyurea, for changing RNA to DNA. That's basically everything we took up until now. Then you have degradation. All right, 1 second. Okay, so after you're done with using these purines, you need a way to degrade it, right? So it can't be salvaged. It's been used for too long, right? You have to throw it away now. You're gonna take these purines, they're gonna go into your intestinal mucosal cells, and then they're gonna form uric acid, and then eventually they're gonna get out of your body through urine. All right. I did not make my own version of this because everything was together here, so I thought it was better that way. Um, um, okay, first you have AMP. For example, again, as I said, I'm biased and I like this more. You have AMP, and AMP has an amino group on it, right? We added it. We added it through the phosphoribosylamine. You have the amino group on it. So you remove the amino group. Okay, minus NH. When you remove the amino group from this, you're gonna have IMP. Okay, over here, as you can see, you're gonna remove the NH3, and then you're gonna have inosine eventually. Mm-hmm. Okay, my bad. It was this one, AMP, and then you have the amino group, and then you have IMP. And for this one, the enzyme is AMP deaminase. Okay, Again, from the name itself, AMP deaminase, all right? AMP deaminase. Okay. Or you just call it ADA. Okay. So for the nucleoside itself too, there is adenosine, there is adenosine deaminase, and there is inosine. All right. Eventually, obviously, IMP is gonna form inosine because you're gonna remove the phosphate. Okay. Now, um, you're basically like going backwards, right? First, you formed IMP, and then you, as a parent nucleotide, and then you had AMP, right? For degradation, you're gonna go all the way back. You have AMP, and you're gonna go reverse. You're gonna get IMP. Okay. Now, there's certain conditions where you're gonna have a problem in the ADA enzyme, right? So here, this it's called severe combined. Wait, I'll just write it here. Hold on. It's called severe combined immunodeficiency. Okay, and it involves again T and B cells. One sec. No, if you if you don't have ADA basically, right? You're gonna have a lot of adenosine, okay? Because this is what's giving you IMP, and then you eventually degrade it. But if you don't have that, you're gonna have a lot of adenosine here, right? Now, as I said over here, wait, over here, you have more DATP, you stop you stop this process of making DNA eventually, because it's like a feedback mechanism, right? You stop making DNA. So the problem here is that if you're not degrading it, right, you have a lot of adenosine, and eventually the AMP present in your blood. And because you have a lot of DAMP present in your blood, you're gonna stop making more DNA nucleotides, basically. And because of that, your DNA is gonna basically stop kind of the DNA synthesis. And uh, she also mentioned that the children they usually die before 2 years. Although I'm not sure that's important. I don't really think so. Anyways, basically the cells can divide. Uh, okay. And then we have mm, okay, and then we have GMP. Okay. Now the GMP. Uh, okay. Okay. Again, you're gonna remove the amino group, and then you're gonna go to guanosine, right? It's gonna go back to what it was basically, and the guanosine turns into xanthine. All right. Now this xanthine, for both the ways, for AMP turning into inosine and for guanine turning into xanthine, after you're done, wait, okay, both the ways for inosine turning to uric acid and for guanine turning to uric acid, you need an enzyme called xanthine oxidase. All right. Now xanthine oxidase over here is going to turn hypoxanthine to xanthine, and then xanthine again, xanthine oxidase to uric acid. Wait, let me just do this one again. Okay, again, you have AMP. All right, you're reversing the thing. You're gonna go back to IMP. IMP is eventually gonna turn into inosine because you removed the phosphate. Okay, you remove the phosphate, inosine. Inosine turns to just its base, hypoxanthine. You see, this is the whole reverse process. Hypoxanthine, and then hypoxanthine has this enzyme, xanthine oxidase, turns to xanthine, turns to xanthine, and then xanthine oxidase again, uric acid. Uh, GMP again, GMP same thing, guanosine, remove the phosphate, okay, and then you remove the sugar, right? Remove the sugar, you have guanine, okay? Guanine eventually forms xanthine again. Xanthine, what happens to xanthine? Xanthine oxidase. Then you have uric acid, okay? Now, this enzyme is also important to remember because this also has a clinical condition with it. Okay, before I go into the clinical condition, because we're going to be talking about it a lot at the end of purine synthesis and degradation. Let me charge my head. 1 second. All right. Okay. Uh, okay. So yeah, I was talking about the DATP here, right? Because the DATP is going to go back, is going to increase in the ADA deficiency, and it's going to stop the ribonucleotide reductase, and eventually, it's a feedback inhibition, so eventually you're not going to have much DNA, right? Uh, okay, gout. Okay, we've all heard about gout, and so what is gout exactly? Gout is basically the overproduction or underexcretion of uric acid. Uh, they're basically the same thing. So if you produce more uric acid, there's more uric acid in your body. If you underexcrete, you get rid of less uric acid. Again, there's more uric acid in your body. So they're essentially the same thing, right? But the more common one is underexcretion. All right. In 90% of the cases, underexcretion happens. Okay. Um, com coming back to this pathway here. There is one form here, hypoxanthine and xanthine. There is one form here that's not very soluble. There's one form here that's not very soluble. Yeah, it's uric acid. Okay, uric acid not very soluble. So this is the problem here. These ones are the soluble ones. Okay. Now, when you have an underexcretion of uric acid and you have a lot of uric acid in your body, you don't want more uric acid to be in your body, right? So what happens is that basically they form. I'll show you. Okay, they form these kind of crystals, which are visible under certain lights, and they start depositing in the joints, and especially in the lower extremities. This is your hand, but lower extremities. So in questions, they usually say, so the person was presented like maybe arthritis or like a swelling in his like big toe. All right. I'm pretty sure you guys have encountered that in the TBL, in the big toe. And like, what is the like diagnosis? Or they're just gonna ask you something about gout. Um, that all right. Keep that in mind. The big toe thing, in the lower extremities. They're gonna have okay. The crystals are called monosodium urate crystals, and they cause an inflammatory response. It's acute and then chronic arthritis. Just remember that it's in lower extremities and it's gonna cause big toe. Um, okay, coming back here again. The most common treatment here is allopurinol. Okay, allopurinol is gonna go with you to second year too, along with methotrexate. So I think that's why both of them are like important to remember because they don't leave your side. Okay, what allopurinol does is you see xanthine oxidase, right? it inhibits this and it inhibits this. So you have xanthine, you either have xanthine or you have hypoxanthine. Now these are like the more soluble forms, so they don't really form the crystals itself. So it's better that we have so it's better that we have um xanthine and hypoxanthine in our blood because then they can get excreted in urine, unlike uric acid, which is first of all not soluble, and then second, it forms crystals and then causes gout and bony arthritis eventually. So remember, allopurinol. One more thing. Wait, what was it? Okay. Anti-inflammatory agents, therapeutic uric acid by allopurinol. Allopurinol. How do you lower uric acid? You inhibit xanthine oxidase. All right. Xanthine oxidase, two steps. Non-competitive xanthine oxidase. All right, another thing. I think this was in our SAQs as well. But it was in some other batch SAQs too. So there is a possibility that it could be in yours. I don't know why I'm predicting questions here. Okay. Um, Now, when you take a diet rich in meat and seafood, you have an increased risk. Why? Because these give you more purines. So these are like purine-rich foods, all right? When you have more purines, you need to degrade the purines more, right? For whatever reason, you need to degrade the purines more, and when you degrade the purines more, you have um more uric acid present in you, right? When you have more uric acid present in you, you have a higher chance of the uric acid going and becoming like crystals, and then going to your joints and then giving you gout. All right. Um, Lower temperature. Lower temperature reduces solubility from chemistry again. They're going to reduce the solubility of urea crystals, again leading them to precipitate. Again, you have gout. All right. Lactic acid compete lactic acid. So, for example, if you exercise and you're like a high risk gout patient, right? That sounds really weird to say. You're a high risk gout patient and you're exercising. So obviously your muscles are going to produce something called lactic acid. Now, when your lactic acid is produced, it has to be excreted, right? But this lactic acid competes with your uric acid. So this is why excessive lactic acid can trigger a gout attack, right? Now imagine you're exercising and then you go for like a meal that's like just meat, right? So that's gonna be like really bad because then you have to like degrade the whole purine thing, but your lactic acid is competing and lactic acid wants to leave first, and then your purine then gets mad and then it goes like, all right, I'm gonna go to your joints and then I'm just gonna stay there. Okay? That's what you get for neglection. Neglection. All right. So um, she mentioned this probenecid here. And she mentioned allopurinol, we talked about. Probenecid is another drug. It basically increases the excretion. All right. So as I said, under excretion is like the most common type of a cause of the crystals. There is overproduction under excretion. So it increases the excretion of uric acid. So more excretion, less gout, right? Fun. Wait, I think I repeated. Oops. Okay. Okay. You have something else called tumor lysis syndrome. Okay. Um, what happens in this is basically when you have a lot of cancer cells are dying simultaneously. Um, right? A lot of purines are going to be released because your cancer cells also had DNA, right? So your cancer cells also had nucleotides and bases. So a lot of purines are going to be released from there. Okay. Now, this is gonna this is gonna go to uric acid. And this is also going to cause hyperuricemia. Again, you have hyperuricemia or high uric acid. Okay. Again, this can lead to gout. So chemotherapy treatment. This is directly from your lecture. In my head, this exact slide is not that important, but you can just be like, all right, cancer patient, right? chemotherapy, like what happens? Your cancer cells have DNA too, so your cancer cells are gonna die because of chemotherapy, and then you're gonna have purine, and then purine is gonna have give you uric acid, and then she can be like, all right, what can you do to manage this? You can give the person or what drug can you give the person? You can give the person allopurinol. Why? Because allopurinol uh, inhibits xanthine oxidase. If you inhibit xanthine oxidase, okay, xanthine oxidase, and then if you inhibit xanthine oxidase, you have the two soluble forms. In this case, also you could use probenecid again because probenecid increases your secretion. So more uric acid excreted, the better you are, basically. So tumor lysis syndrome, again from the name itself, tumor. The cancer is the tumor here. Lysis means breakdown, breakdown of the tumor syndrome. Okay, it's a consequence of chemotherapy. Just think about it yourself. Why would the cancer cells suddenly break down? Because you're giving them chemotherapy, right? That's what you usually give cancer patients. Okay, and then follow your own thought process. All right, if I break the cancer cells down, I have purine. And then again, hyperuricemia. Okay, this one, yeah. All right, this one she could probably ask Leishman syndrome. Uh, this is very important. Um, wait, let me go. This is this. Let's go all the way up here. Should I put another one there? Mm-mm. Here, okay. So when we talked about it here, we talked about the salvage pathway, and I said there is like a clinical correlation with the HGPRT. Um, so there is like a problem in the HGPRT enzyme itself. So you have, let's go back here. Yeah. Okay. So okay. Okay, so what happens here is that you have AGPRT, and what did we use the AGPRT for? We used it to combine the PRPP and the salvaged bases, for example, adenine. All right. Now I got rid of my AGPRT. I don't want to combine my PRPP with my adenine. Now I have like a lot of adenine formed here. Mm. Okay. So I have a lot of adenine, so I have a lot of purine, right? I have a lot of purine in my body itself. Now, um, uh, um, so there's gonna be a lot of bases in my body, and then because of this, there's gonna be an increased de novo purine synthesis. I'm trying to look for the keywords here to show you guys. There's AGPRT, PRPP increased sexual, yeah, causing the increased de novo purine synthesis, and because you have more purine synthesis, you have excessive production of uric acid again, and again uric acid. You don't want uric acid. So I think it makes more sense with here. This is pretty cool. You just have the mnemonic from the enzyme itself. So H, hyperuricemia, because more purine synthesis would mean that you have more degradation. More degradation would mean you have hyperuricemia. Again, hyperuricemia would eventually lead to gout, as I said. And they're usually they're pissed off because the uh, purines basically they cause gout, and gout is very painful. So I don't think anybody wants to be in that sort of pain. So they're usually mad. Retardation, and there's also hypotonia in the muscles. Okay, that was pyrimidine synthesis. Um, sorry, wait. That was purine. Okay, this is pyrimidine synthesis. In pyrimidine synthesis, the base is made first, then you attach the ribose. So in purine synthesis, what you did was you had the PRPP, For PRPP, you kept adding like the nitrogen and then carbon and then all the ingredients slowly, slowly, and then you formed the ring on top of it, right? That was in purine. In pyrimidine, what happens is you make the base first, right? You make the base separately, you make the PRPP separately, and then you combine both of them, right? That's what happens here. And in purine, your main one was IMP, right? In pyrimidine, you have UMP. All right, that's your parent one. UMP is uracil, basically, which leads to CMP and TMP, which is cytosine and thymine. Okay, now in purine, we use something called GAG. We use carbon dioxide, obviously. Now for GAG we need it for nitrogens, right? Glycine, glutamine, and aspartate. In pyrimidine, you do not need glycine, all right? You need aspartate and you need glutamine. Okay? Okay. And then she shows you, all right, glutamine is here and aspartic acid is here and carbon is here. She's not really gonna ask you all of that, because there is much more high yield stuff to ask from. Okay. Now, we're gonna start our pyrimidine synthesis. First, you have um you have let's say you have glutamine here, right? Let me just you have glutamine, okay? I'm gonna show you with the structure, but she's obviously not gonna ask you the structure either. So there's no need to learn the structure itself. But it just makes it easier to understand it this way. When you have glutamine, you add carbon dioxide to it, okay? This carbon dioxide, and then you add a phosphate group to it, okay? This is the phosphate, and then this is the carbon. Now if you go here, this is the phosphate group. Okay, this is the glutamine. Wait, this is a phosphate group. There's a carbon here, and then obviously this is the whole NH2 from glutamine itself. Okay, another thing. Wait, I wrote everything here. Okay, okay. Another thing is that this enzyme here is carbamyl uh phosphate synthetase 2. Right? Again, synthetase, you need ATP, yada yada. Carbamyl phosphatase synthetase 1 was in your urea cycle. So don't mix that one up with this one. If she asks you, um, like what enzyme would you use here, don't mix those two up. Um, I You could just remember it like, all right, we took urea cycle first, so it's carbamyl phosphatase synthetase phosphate synthetase 1, and then we took urea cycle. Oh, sorry, we took pyrimidine synthesis second, and so it's the second one. That's one way to remember it. Yeah, okay. And then, okay, it's activated by ATP, obviously, because the more ATP you have, the more you form carbamyl phosphatase. Phosphate, I don't know why I'm saying phosphate. Phosphate. It's inhibited by UTP as a feedback inhibition again, because the more um the more of this nucleotide you have, you don't need anymore. Basically, it's like, all right, enough. Okay, and this is the regulated step. All right, regulated. This was the first step. What was our first step? We have the amino acid, and we convert it into carbamyl phosphate using carbamyl phosphate synthetase 2. Synthetase 1 was in urea cycle. Okay, step 2. Here we have carbamyl phosphate. Okay, and we're gonna add aspartate. Now here we're gonna make the ring. All right, this is the fun part, making the rings here. Okay, carbamyl phosphate. You add aspartate. Okay. Again, aspartate is an amino acid, and I said amino acids give you nitrogen. All right. So that's why it's here to give us nitrogen because I don't know, fun. So it gives you nitrogen, and it's also gonna help me make the carbon ring. I'm just gonna take aspartate and make my ring basically. Just take everything from aspartate. Okay. Basically, okay. If I take structure, I don't think I'll not show you the structure because it's gonna be a bit hard. But anyways, the carbamyl phosphate is eventually going to form orotic acid. All right, aspartate orotic acid. This is going to be the first nitrogenous base formed. Okay. Do you guys remember what was the first base in purine in water? Hypoxanthine. Yeah, and the hypoxanthine, then you get inosine. Yeah. So this is kind of like hypoxanthine, but in pyrimidines. Okay. You have orotic acid. Now, obviously, when you have the base, you eventually need to form nucleotides, right? Now, this is what we're gonna do here. You have your base. Again, our three friends: base, sugar, and phosphate. Sugar and phosphate together is PRPP, and PRPP just wants to be involved everywhere, so it's back again. Alright? So PRPP is gonna combine with my base, and we're gonna form a nucleotide. Okay? And again, just like purine, it has nothing to do with the name, like hypoxanthine and then PRPP, and then you get inosine. It has nothing to do with the names. You have orotic acid, you have PRPP, you form uridine, alright? They're completely unrelated but related at the same time. Okay. What do you need for this? Obviously, you need an enzyme. So you're gonna have UMP synthase, alright? UMP synthase. This is not synthetase because no ATP. So UMP synthase. Okay. Um, so there is a difference between the structure of orotic acid and UMP. Okay. Uh, Basically, you see, there is like, wait, let me. Okay. Right. All right. This part here, there's like the carbonyl group here. Okay. Okay. It's like COOH. I don't know why this has turned into chemistry all of a sudden, but all right. COOH. I'm gonna get rid of that. Okay, because I need to combine this somehow. And I'm gonna get rid of this part here. These parts need to go away. And I'm gonna combine, when you look at it, look at this. This part here, let me change this. This part here is similar to this part here, and this part here is similar to this part here. So that's, she said it quite a few times that UMP loses the carbonyl group, COOH, this part here, and then this forms CO2 in the process. So that's why I mentioned it. Because I don't think she's really going to ask you about it because it's not that high yield. Because there's so many drugs and clinical conditions that you need to memorize here. Um. Anyways, you formed UMP here, right? Wonderful. Now you're gonna form UDP, UTP. Just keep adding phosphate. Okay. Now, if I said orotic acid was my hypoxanthine, which means uridine is going to be my IMP. Again, IMP was my parent, so uridine is going to be my parent here. Okay, so this is my parent. Parent. Okay, then you have, I talked about this in the very first slide. You basically have like, just say you have UTP. If you add an amino group, you get CTP. Okay, now, uh, and the amino acid, wait, this amino acid, this obviously is going to come from, not amino acid, this amino group is going to come from an amino acid, and that's going to be glutamine, all right? It's providing us with a lot of amino acids. So amino acid, and then you have CTP. Again, you have methylation, the methylation part. Okay, UMP plus methylation, CH, then you have thymidine. Okay, uh, ty ty thymidylate synthase because thymine synthase basically. All right, it's, wait, let me show you. It's over here. Yeah, here. Ignore this one. Here you have uracil, the amination. Okay. Here it says deamination because it's forming uracil, but we're forming cytosine instead. So we're gonna aminate amination. All right. How are you gonna aminate it? You're gonna use what are you gonna use? Do you guys, do you guys remember? All right, yeah, glutamine. There's like one person on. Okay, glutamine. Okay. This is called amination from the name itself, and the same uracil. When you take it to form thymine, you're gonna add a methyl group. Okay. So this one exactly help you remember it completely, especially if you're watching this 2 hours before your final, which I'm hoping you're not. But thymine, there's like an M here, so methylation. All right, that's one way to remember it. Um, so methylation. All right, let's go back to our slides all the way here. Follow the red marks. Where are we? Okay. Okay. In the methylation process, basically, you use tetrahydrofolate again because we can't live without tetrahydrofolate, obviously. So you have tetrahydrofolate here, and then this enzyme here. Okay, and this is the same thing in the picture itself. Okay. Now, clinical correlation. The fun part, obviously. So it's autosomal recessive. I'm not sure if she's gonna ask you. I'm not sure if she's gonna ask you much about this inheritance pattern because that's more of a genetics thing. But that doesn't mean you can skip on it, right? Orotic aciduria. Okay. Orotic aciduria. Can you tell me what you think of it from the name itself? I should have hidden the other parts. Increased or yep, increased orotic acid. Yeah, inheritance pattern. Okay. So why do you think we would have increased uric acid? Because wait, right? Because the UMP synthase enzyme doesn't work, right? Wait, let me yeah, here. Oh, okay. There you go. Okay. So this enzyme is not gonna work. If this enzyme doesn't work, I'm not gonna have uridine, and obviously I'm gonna just keep making uric acid. If I keep making uric acid, I have a lot of uric acid in my body, which is gonna cause uric aciduria. Okay. Now, because you have uric acid increase and you cannot form the UMP because obviously your UMP synthase is not working, then you have no pyrimidine, all right? No pyrimidine, no DNA, right? And there is an anemia that's caused when there is no DNA. Do you know which one? I mean, it's right there on the screen, unfortunately. Uh, it's megaloblast. Yeah, megaloblastic. In methotrexate, if you guys remember, and I hope you remember, um, it was macrocytic, right? Macrocytic had two types, megaloblastic, so it's kind of the same thing, megaloblastic, right? So what do you find in patients with orotic aciduria? You have megaloblastic anemia. It's not curable by vitamin 12 or folic acid. um, buildup of orotic acid or orotic acid in the urine. So you have the orotic crystals they form, and you have the cloudy urine. Okay, and in some patients you have mental and physical developmental delays because you don't have the pyrimidine formed, right? If you don't have pyrimidine formed, your basically DNA is like not forming at all. Not at all is not forming, and then you have mental and physical developmental delays, right? That's why. Treatment uridine. Treatment is uridine. Let me go back there. Well, actually, no. So if I need this UMP synthase, if I need UMP synthase to form UMP, right? And UMP synthase is not working and I have no UMP. What am I gonna do? I'm just gonna give the person uridine. I'm just gonna skip the whole thing and be like, all right, here you go, uridine. All right. I'm gonna bypass UMP synthase. Um oh my god, okay. Software is tricking me. Okay. Now, there is another reaction which involves increased orotic acid. Uh, this one is called, I mean, it's not, it's basically like, eh, yes, it's called the same name, but it's just like when you have a deficiency in ornithine transcarbamylase, right? Or OTC. This enzyme is very important in urea cycle. Um, what basically, I'm hoping you guys did urea cycle. So here we go. Okay. So we have carbamyl phosphate, okay, carbamyl phosphate, right? Carbamyl phosphate was formed at the first with carbamyl phosphate synthetase too. You have carbamyl phosphate, but here we're talking about the urea cycle, right? In the urea cycle, you have this carbamyl phosphate again, all right? This carbamyl phosphate combines with where to go? With ornithine here. Ornithine comes here, and you have citrulline, all right? Now, if you have if you have a deficiency in ornithine transcarbamylase, I'm sure you guys took this, so I don't really have to explain the enzymes or anything, or go in too much into detail. So, if you have a problem in the transcarbamylase, ornithine transcarbamylase, you have increased carbamyl phosphate, obviously, because it's not going anywhere, so it just stopped here. So you have increased carbamyl phosphate. Now, increased carbamyl phosphate is going to increase orotic acid. Okay, why does this happen? Okay. If I keep increasing my carbamyl phosphate, I'm gonna have orotic acid, right? Increased orotic acid gives me orotic aciduria, right? That's basically where okay. The only difference here, I mean, considering the fact that it's in the urea cycle here, the symptoms are different here. Okay, the name is completely the same. It's orotic aciduria. The symptoms are different here. So you have encephalopathy, which is a lethargic baby, like, all right, always tired and comatose, all right. And obviously you have like increased ammonia levels. I'm not really gonna go into the whole ammonia thing because that was in the urea cycle itself. And then eventually there's like a problem with the brain and stuff. So okay. So the main difference between both is gonna be megaloblastic anemia. So remember that you have a type of anemia in nucleotide synthesis degradation. You have megaloblastic and macrocytic anemia, essentially the same thing. So in the or sorry, in the orotic aciduria in nucleotide synthesis, you're gonna have anemia, megaloblastic anemia. In the urea cycle orotic aciduria, you're not gonna have an megaloblastic anemia. So that's one of the differences here. Mm, okay. I think this. Oh yeah, it's basically the same thing. Glutamine. This was where we basically put all of our carbon dioxide, ATP, whatever. Then you had carbamyl. Let me just. Okay, let's revise it here. Then you have carbamyl synthetase 2. Carbamyl synthetase 2 gives you carbamyl phosphate. Carbamyl phosphate gives you orotic acid. Orotic acid has to form UMP, which is going to be our parent nucleotide in the pyrimidine synthesis, right? Okay, so To form UMP, you need something called UMP synthase. Okay. Now, there is one problem with UMP synthase. Okay. If you have no UMP synthase, you have orotic aciduria. We just went over it, obviously. More orotic acid, and yeah, that's basically it. And then UMP, you have CMP, and then you have TMP, which is cytosine and thymidine. All right, that's basically it. This is in nucleotides. This is how. This is where you stop, and this is how you have orotic aciduria and nucleotide synthesis. But in urea cycle, this is from the urea cycle. You have carboxyl phosphate, you have citrulline, anything. Carboxyl phosphate combines with ornithine gives you citrulline. And if this enzyme here stops, you have carbamyl phosphate, and you can see that how carbamyl phosphate eventually forms orotic acid. So both of these eventually lead to orotic aciduria. Two separate lectures, orotic aciduria. I think that tells you that it can be in the exam. Just saying though. Pyrimidine synthesis. Okay. More. Thymidine synthesis. Thymidine, as I said before, thymidine is only used in DNA, and U, uracil, is only used in RNA, right? Enzyme is thymidylate synthase, all right? As I said, that when you have UMP here and here, you have methylation, right? When you have methylation here, you're gonna require THF, and you're gonna require THF for methylation. And you're going to require this enzyme here, thymidylate synthase. It's pretty obvious from the name. You're synthesizing something related to thymine, and obviously it's going to be TMP, right? Then you have regeneration of THF requires, okay, we went over this. THF requires dihydrofolate reductase. So folate, THF, and then THF, right? Your methotrexate comes here and goes like, yeah, no more THF for you guys. This affects both the human cells and the bacterial cells, cancer cells, basically. The bacterial antibiotic was sulfonamide. Yeah. Thymidylate synthetase is targeted by anti-cancer drug, uh, uracil. Okay, so here. This is another analog like methotrexate itself. So, thymidylate synthetase was used to make the TMP, which is used in DNA. Now I'm going to stop the synthase, and because of this there's not going to be TMP. If there is not going to be TMP, there is not going to be DNA. That was basically it. I think, yeah. Wait, let's go over this one more. Let me show you the differences between both of them. Okay, wait. Um, where did it go? Mm, okay, I am not done. Wait. Okay, yeah. Just 1 second. Let me just download. All right. Um, this is basically kind of like a summary. Okay. How do I write this? Okay. All right. So first of all, you used we're gonna start with purine. Okay. This is basically a summary of the entire thing. Um, After this, I did add questions, but there is just one person here. He's probably gonna leave too soon. So I'm gonna put the questions in the slides itself, and I'm gonna put the explanation down for whatever is needed. And I think most of them are pretty straightforward. Again, as I said, it would be better if you focus on methotrexate and gout. I'm not saying don't study the entire thing, but just in case if you're watching this right before the exam, which I'm hoping you're not. Look at methotrexate because there were a lot of questions on it. Alright. For example, um, let's say, wait, let me take the questions out. Alright. So I think this was our SQ, and they had like a whole case scenario, and it said, there was a severe pain in his right metatarsalpharyngeal joint, which is again the big toe pain, and I said the big toe is the gout thing. Alright, what is most likely the cause? Gout or deposition of monosodium crystals in the joint. That was it. And how does a diet contribute? Because I said diet can cause gout itself. For example, if you have meat or red meat or even lentils that is like rich in purines, that's gonna increase your purine degradation. And if your purine degradation is increased, that means you have more uric acid in your body. More uric acid, more monosodium crystals in the joint. All right. Um, that was there could also be like they can also ask you how would you what would you see in the synovial fluid analysis, all right? Which would be like, all right, this person has got. You would see the monosodium crystals. This is just entire thing again. Um, um, for methotrexate, they can ask you, methotrexate is an inhibitor of what enzyme? Do you guys remember what enzyme? Okay, interesting. Oh, yeah, okay, yeah. It's dihydrofolate reductase. Yes, correct. Um, so that would happen. And then if you have, for example, treatment with methotrexate and you have side effects, then what are you going to give? Because methotrexate is like you're stopping the folic acid is being affected. You're going to give the patient more folic acid, right? Um, that's like there were a lot of questions on the drugs and their things, especially like sulfonamides, methotrexate, more methotrexate, and gout itself. Yeah, but they're at the end, and then if you want, you can look at them, obviously. Anyways, we're almost done with this. We're gonna go to the summary now. All right. Now in purine, I said you have, again, I've run this so many times. I'm hoping you're drawing with me because it's really gonna stick with you. You have ribose and 5-phosphate, and then you have PRPP, right? ARPP. Ring built on, right? The ring is directly built on ribose in this one. That's one of the that's one of the main difference. You keep adding nitrogen, whatever you want. You want nitrogen, carbon dioxide, whatever you want. You're adding it to your PRPP, and you're forming the molecule on top of PRPP, right? So you're not making it separately and then joining it like in pyrimidine. All right. The key enzymes are PRPP amidotransferase. Do you remember where this was used? Hoping you do. You have PRPP amidotransferase. You're transferring an amino group, okay, to PRPP. So you're making mm, phosphoribosylamine. Yeah. Okay. Okay. Phosphoribosylamine. Good. Phosphoribosylamine. And then we skip a bunch of steps. And then you have IMP. And from IMP, wait, this is not. Alright. Oops. IMP. Where do I write this? Uh, oh, how do I get rid of it? Okay. Clear out my drawing. Okay. You have IMP, and then you have IMP. You form GMP, and you form AMP, right? And you need the enzymes there as well. That's the main thing in purine. Uh, And then you have pyrimidine synthesis. In pyrimidine synthesis, you have something called, hmm, you have something, what do you have? Yeah, the ring is made by a cell, but what's it called? Uh, yeah, okay, well, okay. I think, yeah. Eventually, yeah, you form carbonyl phosphate through carbonyl phosphate synthetase. Two carbonyl phosphate synthetase. Okay. Alright, for this one, I'll go back and explain again. But anyways, carbonyl phosphate synthetase, then you form. Oh, where did it go then? Okay. Carbonyl phosphate, and then you have orotic acid, and then from orotic acid, you form UMP with UMP synthase, right? By the way, the starting was an amino acid. Wait, okay. Orotic acid. Orotic aciduria. This was the clinical part. Need to fix this. All right. Um, this is the key regulating enzyme. All right. So remember this enzyme. This enzyme and UMP synthase. All right. Carbonyl phosphate synthetase and UMP synthase. All right. Uh, okay. In the purine thing, this one is the key enzyme because this was the regulated stuff. All right. Okay. Initial nucleotide formed. This is the parent nucleotide. This is supposed to be UMP. Yeah. I'll fix this and I'll send it again. You don't need to know this. Over here you need GAG basically. You need CO2, formal THF, and you need ribose. Aspartate and you need lysine. End products AMP, GMP. Sorry, glutamine. Here the difference is that you need lysine here and you don't need lysine here. Okay, anyways, AMP, GMP, you have UMP, CTP, DTMP. This had a specific enzyme. Thymidine something synthase. You don't need to know the exact name. Thymidine something synthase forms DTMP. All right. There is a drug that targets this enzyme, and because of that, you have less DTMP because it's an anti-cancerous drug. So less DTMP, less DNA formation, less DNA formation. Cancer cells can't use it, and so your cancer cells are dying. Basically, anti-cancer. Okay. Regulation, feedback inhibition by AMP. AMP and GMP are the main products of this. So if you have enough products, you're obviously not going to need anything else more, right? So you're going to be like, all right, stop. Feedback inhibition by UMP. It can also be by CTP and TTMP. You don't really need to know this. This in uh, in they both take place in cytoplasm. In CPS, I think in urea cycle you learned that it happens in the mitochondria. So that's the difference between CPS1 and CPS2. I don't think she's really going to ask you the differences between them, but it's to better. I think you should know honestly that it happens in the mitochondria because I think in the urea cycle you learned about it in the mitochondria itself. All right, I'm just gonna go over pyrimidine synthesis one more time because I think we've gone over purine synthesis a lot of times, so it should stick with you by now. I'm just gonna go over purine synthesis one last time, and then we can end this thing. Sorry, pyrimidine synthesis. All right. Dun. Okay. Uh, okay. First of all, this is not needed. Glutamine. All right. What are you adding to glutamine? Glutamine gives you the nitrogen part, right? Nitrogen. You need a carbon part too, right? You're gonna get that. here. Okay, you need the carbon part, you're gonna get it from here, and you need phosphate. Okay, and you're gonna get phosphate from there. Okay, glutamine to carbamyl phosphate. Okay, you're synthesizing carbamyl phosphate, so carbamyl phosphate synthetase because ATP. Okay, and UTP because feedback inhibition. You have enough end product, so you don't need to make it more. So that's it. All right. Glutamine, carbamyl phosphate. Carbamyl phosphate, uric acid. You use aspartate here. Okay, it gives you the carbon ring and the nitrogen. I don't know. In my head, aspartate and uric acid are kind of like, it just sounds kind of similar for some reason. Because like aspartic acid, anything that ends with tate, by the way, is acid. All right. So if I say orotate, it kind of means uric acid, but nobody really says that. So one second. Okay. So you're taking an acid to make an acid, basically, right? Carbamyl phosphate, aspartate, uric acid. Now, uric acid is our base, like hypoxanthine, and this is gonna give you uridine. Uridine monophosphate. How is it going to give you uridine monophosphate? This is your base. You need the other two friends. Your other two friends are PRPP because it likes being included in every single thing ever. Alright? Narcissistic. UMP synthase, you have uridine monophosphate. Uridine monophosphate, keep adding phosphate, UDP, UTP. And then the UTP is going to be converted into CTP, or whatever, and then CTP and thymidine, TTP. Alright. If you add an amino group, okay, amino group, CTP, methyl group, thymidine. Okay, and for thymidine, you need THF, right? Again, remember, T. Just keep correlating stuff. Thymidines, and you need this enzyme, important enzyme, because there is a drug that inhibits the enzyme. Okay, uric aciduria. What happens in uric aciduria? Caramel, no, not here. UMP synthase, more uric acid. More uric acid, uh, happens when you have more uric acid, you have uric aciduria, and you have its own symptoms. Remember, megaloblastic anemia. It cannot be cured by by B12 or folic acid. How do you treat it? By uridine. Just bypass the whole thing. Who cares about uric acid giving you UMP? It's overrated. You can just give them uridine yourself. Okay. And then, yeah, this is the urea cycle one. Both of them have uric aciduria. They have different symptoms. Main difference is anemia. Here you have this combining here. More carbamyl phosphate, more uric acid. And then, okay. That's basically it. Um, just, that's basically it. Thank you for listening to me. I'm sorry for the multiple pauses. And I hope you benefited from this. Again, focus on what I said to focus on, but also look at the slides and don't skip anything unless, again, this is right before the exam. And good luck for your MOL final.